So, hi everyone, and welcome to today's digital information meeting and Q&A session with Gabather. Today, the chairman of the board and the management team will be providing you with an overview of Gabather's current situation with reference to the ongoing rights issue and the plan going forward. They will also be updating you on the latest results from the ongoing target engagement study of GT002. And we will leave time for a moderated Q&A session at the end, and you can submit your questions in the chat. You will see that the presentation will be in English, but you can submit your questions in both English and Swedish. We'll take as many questions as time allows us to and end the session at 2 p.m. So I will now hand over to the team to introduce themselves and start the presentation.
So, I'll introduce myself first. My name is Mikael Owenvid. I'm the CEO of Gabather. I've been at Gabather for the last almost 5 years as a CEO, and before that as principal scientist. And I think most, if not all of you, are familiar with me, either via email or previous presentations. So I'll keep it short and give over to the next person in the management team, which is Stefan.
Yes, hello, my name is Stefan Rönnmark. I joined Gabather in 2016. My background is I have a PhD from Wenner-Gren's Institute. I've been working with in drug discovery and development for the last 25 years, and I really enjoy this work. So that's short me.
And that gives over to Christine.
Hello, I'm Christine Ryan. I'm the Chief Operating Officer at Gabather. My background is within neuroscience. I did my PhD at the University of Cambridge and then worked for Big Pharma, AstraZeneca, for many years. I've also worked with smaller companies, actually CROs, so contract service companies. An American one that happened to be in Asia. And I came to Gabather just over 5 years ago in 2017. Yes, and I'm very much involved in the clinical trials and ensuring that we get the best possible studies that we can get done. So right now we've got a very nice study ongoing, which is, of course, EEG and fMRI. So that's me in a nutshell. Thank you.
Well, my name is Anders Lönnberg. I'm the chairman of the board. My background is I used to be state secretary at Ministry of Health and Social Welfare. I've been in Big Pharma for a number of years, and also I've been the working for the government as nationally with the life science sector as a coordinator for life science. So when we started here, I would like to say I hope most of you have seen the press release from this morning about the latest results from Portugal. Into a sense, I think to many of you will be a repetition today about what Gabather has done and is doing and our plans. But then again, we all know that repetition is the mother of knowledge. Unfortunately, it's also the father of Alzheimer's, but since that belonged to our sector, we're trying to fight that, aren't we, Robin?
We are on the way. Right. Thank you, Anders. I almost forgot your names. Talk about Alzheimer's. Just kidding. So I will talk you through our presentation today. It will take about 20-25 minutes. And in this presentation, in contrast to previous ones, which many of you perhaps have heard, I will be focusing a little bit on the data and on the way we get the data and trying to tell you a little bit about our philosophy and what we're actually trying to achieve, and our ideas behind the GFMI study, and specifically, but that's of course not to say that we'll not do other things at Gabather too. So if we start with I will go through the rights issue really briefly. That's the acute occasion, but also I will go into the background and the roadmap. And of course, then we got the question and answer session at the second half of this meeting. So we'll go for the next slide. I think I think the key bullet point in this emission is actually that we want to extend our runway, so to speak, for continuing the clinical trials, and of course the licensing negotiations, which we engaging in here for the next year to 18 months. This is what the funding is actually for. And of course, it is actually a relatively smaller issue with the preferential rights. This was assessed to be the best option for Gabather and the shareholders, we think, and it's not reaching for the stars, but at least reaching for the sky for us for the next 12 to 18 months. And we are quite confident that we'll be okay. If we go to the next slide on the status, I will of course go very much in detail on the phase 1b to A, and also a little bit of semantics around that, where the clinical trial really is, and the target engagement study, which is the key actor in this particular show. The patent series, we're actually doing quite a lot of work done by Stefan on the patent. We have, I think, a very interesting patent strategy. We've expanded it. We started with looking at composition of matter patents. We expanded it to synthesis patents, extended it that further into formulation patents, and finally into medical use patents. So layer by layer, we've added IP value to our portfolio. And of course, we should not forget the pipeline compounds. And I put them basically in at this presentation because they tend to fall away because of the focus on GT002 and the results on the clinical trial. And I always have to repeat that we are actually still a bench-to-bedside company. We synthesize our own compounds based on our own model of the molecule and put them all the way, at least for GT002, to the bedside of the volunteers that we're currently trying on. So that would be the snapshot status. So if we go to the next slide, and this is our milestones we got. Again, as I said previously, there's a lot of focus on the clinical trials, and it makes a lot of sense because I think one of the absolute key moments for Gabather was when we transitioned from our preclinical to our clinical trials. I think it was absolutely key. And what you're seeing here, milestone-wise, is the clinical trials. And here the key take-home message is that we've got positive outcomes since 2019, from starting with a single ascending dose study via a multiple ascending dose study to now the third clinical trial in healthy volunteers, where we look at EEG fMRI, which is the focus of this presentation of today, actually. So if we go to the next slide, our clinical phase is ongoing. I'll talk about this in a second. We have the phase 2 studies, which are actually following up on the data that we're generating from the phase 1, and I'll talk about our key lock principle. I think it's very interesting approach actually that we're using. Not to forget, we do have our GT compound pipeline, and we do have actually data on this compound, which we're going to release also during this year of work we've already done regarding also the second patent series. Never forget that GT002 is one member of one patent family, and we have sort of not mistreated but sidelined the second patent family, and there's a lot to get there too once we actually have the resources to do a proper preclinical workup on this particular series. So let's not forget that one. And the immaterial protection, IP protection on the key markets, there we've really been quite thorough. We've been also taking some markets that might be not that relevant, but certainly we are always working in Europe and US regarding the IP. And our IP Council is actually an office in Washington, D.C. we worked together with for many, many years, and we're actually quite happy with what we've come up with. And again, I'm repeating myself here, but important for us that the pattern strategy of a layered compound where you start at the core with a composition of matter and then you pattern the synthesis, which we actually improved quite a lot to formulation and to medical use in the end. So we actually literally go from chemistry to medicine and the patterns. So that's actually, I think, a very nice strategy. So if you look at the next slide, and this is a slide I actually usually take out. This one is a very nice one that Stefan made for me, and it was to show you what the EEG is about. EEG stands for electroencephalogram. So we're looking at electrical recording of the brain, and the brain is if you put it very crudely, of course, an electrical organ. If you put a cap, like on the left-hand side of this picture, and you see the currents that come in the brain talks to itself, and different parts of the brain talk to itself at different frequencies that you can see on the right-hand side. And this is the kind of recording that you can do, and you measure the electrical activity in the brain. And we are, of course, as drug developers, we're interested in looking at the modulation of that activity in very short time sequences, in seconds, in the brain, modulated by our drugs. That's what we want to do, and that's what this talk is going to be about, and present some data about this and why we think it's a good idea. So we're using EEG, as in the title, as a biomarker. We do not use, as you would use biomarker in a study looking at insulin and blood glucose, we use EEG as a biomarker for mental activity in the brain. And in my opinion, it's a very advanced, very sensitive, and also, don't forget, it's very non-invasive, because you literally just put on a cap and a recording device to get your data out. And in the next slide, I can see you can see that I've put a big red dot there in the middle, and a lot of arrows coming from it. And the thing that I want to highlight here is that what we want to do, we take the EEG as a center. That signal, together with the fMRI, is our biomarker, and the EEG pattern that you can see in the different disorders that we might be able to treat with GT00 is at the very center. So each of the disorders, be it schizophrenia, autism, depression, will have its particular pattern, and that pattern we want to match in a key and lock approach with a pattern that we can induce with our drug, which is what we're measuring right now. So on the right thing, this our approach has been, we look at the pattern of GT002 in healthy volunteers. We know what the pattern is going to be in the different disorders on the left-hand side via the EEG, and that is what we're trying to actually address. So then GT002 can be tested then in patients, and that's then our next step following our 3 clinical trials in healthy volunteers. So in the next slide, I don't know if I already hop in. This is an example of how do you look at the different patterns in EEG. If you look at the left-hand scatter, you have different frequencies, and you compare on the row to the left, a normal, and the row to the right with a mental disorder. And you can see on the heat maps, the different activities, the difference in the activities and the different frequencies between normal and mental disorders. And once you have such a pattern, then you know what you want to target with your drug when you want to restore the activity pattern so it matches a normal or healthy volunteer compared with the mental disorders. And what you measure is actually the power, i.e. the intensity of the electrical signal in the EEG. And this is data that we had, I think, a press release a couple of weeks ago, and we're actually still looking at it. And you can see that there are differences on the right-hand table here in the blue, dark blue, between the activity in the normal and activity in the disease field. And you can also see by the bars that the disease does not mean diminished activity. It can also be enhanced or exaggerated activity at certain frequencies, and it can be diminished activities in other frequencies. So for example, beta is activated in disease and delta would be diminished. So if you then take the next step, and now we're looking at GT002 and looking actually at what is called pharmaco-EEG, i.e. you're looking at EEG patterns of pharmacological agents. Again, you look at the right-hand column here, you're comparing a normal with a mental disorder disease that we have looked at before, and what you want to induce with GTX. And this is an example. I'm just trying to clarify what we are actually trying to achieve what you want to do is you look at the right-hand column as the different brains from the different frequencies that you want to restore what was a normal as compared to a mental disorder. So if you then compare, say, on the top row and the delta row, you have a normal, you have a diminished activity in the mental disorder, and what you, of course, when you want to get the activity back to normal after giving the drug GTX on the right-hand column. You will not achieve this, of course, in all frequencies and in all disorders, but it's the goal and the strategy that we've chosen to do. So the idea is to restore the brain activity, e.g., from an disordered from an unhealthy to a healthy pattern, and that is what we mean by a key lock principle. So you're giving a certain pattern in form of the disease, and you want to have the pattern of the EEG has to match the one in the healthy volunteers. So set it back. And I think this is an animated slide. If you click one more time, you will maybe see. No, it's not an animated slide. Okay, then we go to the next slide. Summary of our successful, and I've already preempted that our third phase 1 study is also successful. So the key insight is we have an orally available capsule, and it might be very trivial because people can say, well, what's the big deal? It's a big deal to have an orally available CNS active compounds in a capsule formulation that we have. And if you go around, there are not many people that can give you a clinical trial material to test, for example, in the formulation that we have. It's very well tolerated and safe. We have not seen really any deleterious side effects in all the volunteers we've exposed them to our material. We've also been successful, again, something that seems very easy, but it's not, the upscaling of the clinical trial material, so we can produce more clinical trial material. We have been shopping around, so to speak, and Christine has put a lot of work in finding the right suppliers, and we do have the material ready for clinical trial, and we actually developed our first product form. So that's actually a proprietary way we developed our drug. It's nothing that we've taken off the shelf and just put it in. We actually developed and actually patented ourselves. So that would be the key insight or snapshot on where we stand now with GT002. And I think if we go to the next slide, I just wanted to show you, I talked about a little bit about what is our philosophy, and the more I've been working with this presentation, the more I got fascinated by the concept of a window into the brain with EEG and fMRI. EEG stands for electroencephalography. fMRI is an imaging technique, which stands for functional magnetic resonance imaging, where you look at activity on the brain during an imaging, while fMRI, you have the volunteer, you can see it in the picture here with a cap, and we're doing this simultaneously. So we're looking at the activity with EEG, and we're looking at where in the brain that activity happens, and I'll go to that later too, into in the brain modulated by GT002. So that is what we have been spending quite a lot of time. It's quite it's not technically unchallenging, to put it very, very modestly, because you're actually getting signals from both EEG and fMRI in one machine and one patient at the same time. And you can imagine this isn't all this electrical currents you're measuring. You don't have to be an engineer from KTH to know that this is actually quite tricky, and there's a lot of data processing in it. And when I found the concept of window into the brain, I thought this is really smart that I just invented this, until I found out that actually a window into the brain is actually very common, and for fMRI has been around for a long time. So I found a review article where they use exactly that concept. I think it's a very nice one. And I'm going to show you why in the next slides. If we go to the next slide, this slide is actually real life data that we got very recently from our partners in Portugal. And the reason I put on this slide is just to show you that it is not a question of pushing somebody into a big tube and then getting the data out on the computer. There's a huge amount of data processing, and this is not something you can do with an algorithm or AI. This actually you have to do with a hand. You have to correct for the slides. You have to correct for the motion of the head. Where are you getting your data from, slice by slice? And in fact, brain region by brain region, and I come to that later. So-called pre-processing, you have to smooth, you have to filter with. For all of you that are big fans of data analysis and signal interpretation, and that doesn't include me, it's a huge amount of work. End of the day is that you do extract the activity of the brain that you measure in fMRI in certain brain regions. And you know which brain regions are responsible for what kind of activity in a resting state. And there's also a difference between activate and resting. So that's what RSN stands for, resting state networks. The brain works in networks, i.e. different parts of the brain talk to each other and get activated dependent on what task or what the situation you're in, if you're thinking, sleeping, or engaging in cognitive processes, which we have been interested in from the very beginning from GT002. And if I hop in now into the real nitty-gritty of the next slide, this is an example of the networks we look at, and this is data from our study actually, where you look at the yellow areas. These are slices through the brain, and what you can see the areas that you measure in, and it's from there that you look at the activity, either increase or decrease in activity in so-called different networks. Brain, again, works in a system of networks. Different networks are responsible for different physiological activities. So the dorsal attention network, for example, that I've put up here as an example, we're looking in total of 12 networks. And if you think of the slide before regarding data analysis, you have to go through each networks individually and through each individual individually and through each drug individually. So it's a huge amount of data processing that you have to do. So each network, again, has certain functions in the brain, and the salience network. I know, of course, all of you are wondering what the salience actually means. I looked it up myself. It stands for important or predominant network. And this is an important network, which again controls other networks that are important for emotional processing and cognitive regulation. Again, these areas that we're interested in. So we take a look at these networks, at the activity in these networks, we measure it, depending on which drug we're testing, and then we get the data out in the different networks. I put some names on. This list is not complete, but we're looking at least 10 different networks. They basically regulate almost all activities in the brain, including, for example, language, auditory, visual networks, and so on. So if we go for the next slide, and here I've given you an example. How does it really look like when we look at the data in a nitty-gritty way? It doesn't get more detailed than this. Here you're comparing two medications, group A and group B. I've taken them out of the data set that we have received. And what you do is you take each dot as one healthy volunteers, you give them the medication, and then you look at the activity in the different areas of the brain that I just mentioned, the different networks after 2 and 4 hours, and you can see if the activity increases or decreases in the fMRI signal. So that is where we are now, and we can, I think, quite confidently say, we can discuss it later too, that GT002 has an effect. So regarding the data and presentation, the data, the plots on the right-hand side, they are representations of the data that's on the XY axis on the left-hand side. So you can imagine that there's a huge amount of data to go through, even with the number of volunteers that we have now. But it's, in my opinion, the most sensitive way to look at an activity of a pharmacological CNS agent in the brain. And after the meeting that we had with our partners, I must really say that this concept of a window into the brain is actually holds true. And I'm very much convinced, more convinced than when we started the EEG project some time ago, that this is the right way to move forward to detect activity in the brain. So I think I do have another slide regarding the data analysis and the EEG. And this is the default mode network, where we saw some interesting results. And there are definitely differences there. This particular network, again, its physiological relevance is quite high. And it regulates the internal modes again of cognition, and that is the area that we have to go back to the cognition deficit in Alzheimer, which Anders mentioned at the very beginning, is of course relevant for us. So that's the way we can actually find out which areas of the brain are regulated by GT002. And once we know that, to go at the beginning of our philosophy is, of course, which areas on the brain could be then interesting in a disease state, because we know that our ligands targeting the GABA-A receptor system, the GABA-A receptor system is involved in almost all neuropsychiatric disorders, but we have to find out which ones. So this is a nice way of looking at activity modulation in the brain. And I think this is all the data presentation I have. I do have a couple of more slides, I believe, because I think I've lost track of the time. So my temporal network must have, here we go. And this is an outside, and here we just show how we've been on this track, I think, from the very beginning and for quite a while, of course. For GT002, which is our candidate drug, which we have pushed through all the phases of drug discovery, which you can see, and now we are at the borderline between 1B and phase 2. And this, I just want to mention very briefly, phase 1 generally is only healthy volunteers, and phase 2 is patients. But in our case, we are actually, the data that we're showing, we're looking at the effect of a drug. So we are very much on the borderline between a phase 1B and 2, because we're looking not only at safety, we're not looking only at tolerability, but we're looking at what effect does the drug have in the CNS. So we are very much on the borderline, sort of on the doorstep of phase 2, in my opinion. But that is maybe a semantic question too. And as you see here, indication 1, 2, and 3, that's exactly the stage where we are now with the EEG and fMRI data. We're hoping that this can then guide us, if we are applying key log principle, to which indication to move forward to with the biggest chance of success in the clinical trials. And I was asked, and I gave a very simple example that, you know, we are testing this as if it was a medicine against high blood pressure. Well, you would test it in healthy volunteers, and you will only go ahead with compounds that actually lower the blood pressure in healthy volunteers, because then the chances that you have a good success in people suffering from high blood pressure is much higher. It's not more complicated than that, actually, in the basis of it. And on the bottom left-hand side is the GT00X, which stands for those compounds, both from the pattern family of GT002 and our second pattern family, which are only in paraphrasing here, but it's at the level towards CD selection, at least towards preclinical, and there's a lot of potential to be able, with the resources, to move these compounds forward. Again, remember that this is a bench to bedside operation. We do have the compounds ready, and we do have the model to make more of synthesize more of candidate drugs at Gabather. It's not a one-trick pony, which I've been also asked previously. And I think I'm at the very end of my presentation here. And of course, bottom line, what can you actually do with this kind of project? What is the potential in there? And it's very much a number of millions and a billions game. The key thing here is that, of course, target engagement is something that was required and requested by big pharma and small pharma partners that were talking to us. So I think they will be more than happy. We're on the radar of most of them with the information that we are gathering now. And this, of course, our reference point, or the one that I'm always quoting, is the success that Biogen had with Sage Therapeutics. And that is because this is a small molecule also targeting the GABA-A receptor system, different in molecular structure than ours. And I'm going to be a little bit cheeky and say I think our compound has better molecular properties, but don't tell that to the guys from Sage. But so they made an agreement with Biogen, I think clinical stage 2 or maybe to 3 for 1.5 billion, 1,500 million. deal agreement with that molecule between Biogen and Sage. So I think that is roughly, the framework within the potential that you have if you succeed with this kind of compound in an interesting indication, and the indication that we are working in, again, to go back, if you think that you can have a compound that modulates cognitive networks in the brain, and if you think about the potential, not only Alzheimer, but just any other cognitive defects, the potential is tremendous, I think. Never mind the fantastic million numbers. I think it's really great, and I'm very happy about the data that we've got, also relating to the press release from this morning. So I think, I don't know if this one is also animated, but I'm next to finished, I would say. Unless there's another slide, no, there we are. I think there should be one last animation, because I do believe I wrote politely thank you on the Gabather slide, but that has disappeared. Sorry about that. And then I just want to pick up, there was one very kind shareholder that said, Robin, never forget how far you've gotten with such a small team. And that's the guys and girls to my left and to my right, and including, of course, Anders, Stefan and Christine. And remember that for 3 people to move this so far is actually not bad. So sometimes when the data doesn't look so good, I always lead myself on that fact. And I think we've gone quite far. And I think with the data that we've got today, we can get even further with our strategy. This is the right one. And with that, I think I've lost track of time and space, I think, but signaling is I'm good in time. So I thank you for your attention, and you'll have to take it just through your auditory system. That I thank you.
Great. Well, thank you. So as we said before, we'll now move over to the Q&A part of the session. And just to clarify that if you have any questions, you can write them not in the chat, but in the Q&A/F&S function, which is on the top right of your Teams channel. So yeah, you can write your questions in English or Swedish, and then we'll cover as many as possible during these remaining 30 minutes. We have got a couple of questions already, so we'll kick off with them. And the first one is, the news that we got this morning was very pleasant to get to know of. Will this now be presented to potential partners? Are we first that one?
I'll be the loudest guy in the room, as usual. Definitively. I mean, this is actually the first question that will come. Remember also that the reason for engaging in a so-called target engagement study was because that was specifically requested by the potential partners. We want to see that kind of data in order to move ahead. So of course we're going to give them this data, and they're already asking us for this data because they're also for the press releases, of course. So that is the thing that we've put ourselves on the radar and said, this is what we're going to do. So they know that we're going to and this is the data that we're going to show them. Absolutely.
Very good. We've also heard in the past about different diseases which GT002 could be a potential candidate for when going into phase 2. Is there any change of direction with the news that we got this morning?
Well, I'll give this question to Christine, Stefan.
This means that, of course, we seem to be in the right direction, and the diseases that we're most interested in happen to be schizophrenia and depression. But of course, this even widens our possibility. But we haven't actually got preclinical work so far, but otherwise we would consider maybe ADHD, for example. So it actually widens our horizon rather than diminishing it, but we're still absolutely on the same track with the same diseases of interest.
Very good. The third one is, we have earlier heard about Kastanenka Lab. Is there any news about the collaboration with them?
We need soft funding for that. We can't, we're very interested, and Xenia is also very, very interested. So we have that very good contact with that lab, and we have the preliminary results. And we did apply actually to the Alzheimer's Fund, Discovery Fund in the United States last year. We haven't applied this year because, of course, the funding there is allocated to different types of collaborations. And at that stage, we were looking for preclinical, but we hope to be able to continue applying this year and next year, depending very much on their calls. We can't just apply to calls that are not appropriate.
Yeah, maybe I can just chip in here that, you know, the Kostanenka lab in the States and so on, they were hit very hard with Corona and Corona restriction, because these are lab active activities, and they've been actually hampered quite a lot.
Very good. Fourth question here is, as a shareholder, I also think of communication outwards in the company. Will there be any changes around how Gabather will communicate to get the company to be more well known in the public, to the public or potential partners?
I think, I mean, with the new strategy that we have, actually also engaging in a communication bureau, we will definitely increase our communication and also do it more frequently when we have news to actually communicate. But we are actually consulting with a very nice communication firm, and this will actually hopefully change our information flow to the shareholders.
Perfect. If there's nothing to add on those questions, we have received a couple via email. And the first one is, what are the main reasons to invest in Gabather, would you say?
Well, I guess that question goes to me. The main reasons for Gabather is, well, basically, it depends. If you just listen to the presentations that we gave you, and the road and the potential that is in this kind of projects, with the team that is behind it, I think that's good enough reasons to invest in Gabather. It is many people actually invest in Gabather not only for purely financial reasons, but also for interest in developing new medicines. And I can tell you that when I walk around and talk to, for example, patients' unions and associations, their first question is, well, when will you guys come up with something new that we can treat our whatever relative with? So the market is there if you want to invest in Gabather. The unmet medical need is enormous and is increasing, and Gabather is basically a shot on a goal that is enormously rewarding. I think that's why you should join us. If you're interested in exciting journeys, you should definitely join.
Very good. And second one, what are your long-term plans?
Long-term plans for Gabather is actually, again, we go back to bench to bedside. Long-term plans, medium-term plans is to get a licensing agreement with activities that we have now, with the revenue that we generate from that. Then we can develop our pipeline and become a proper drug discovery company based on CNS. I think the potential, as we have seen there in just the number of indications, is enormous. The number of candidate drugs we can generate is very, very large. So once we've gone over, say, the first hurdle of getting a licensing agreement or a partnership or a joint venture with Big Pharma, then we are on our way. So the long-term strategy is to become a properly established drug discovery and development company within the CNS area.
Good. And what information can be expected in 2023 during the year?
So the information that will definitely be communicated in 2023 is the completion of the target engagement study, the EEG fMRI data. which is very exciting. And I mean, we just got the tease of what this could actually mean to the company. And I think also, in a sense, this increases our chances also to finding a partner and also getting sort of a proper collaboration further ahead with GT002. So that will come, and we are also working on the pipeline compounds. So this is sort of preclinical studies that we are conducting, and sort of in the selection of which compound to move forward into clinical trials. This will come towards the end of this year, and most likely we will also move forward, or we will move forward with the GT002, identifying the indications that we are planning to go into patients, doing a smaller phase 2b study in most likely in schizophrenia and possibly also depression. And with the background of the data that we received, I think the potential for some good effects in patients are increasing. So this is the information the shareholders can expect to get this year.
Good, thank you. Can you tell us anything about the other compounds in the pipeline?
The other compounds are both from the GT002 series. They are similar in structure, but they have different properties. And they, as you may know, that GT002 is a positive allosteric modulator. We have other compounds in this series that are also positive allosteric modulators, but we also have others that are negative allosteric modulators. And in this sense, they have different effects. And we haven't decided which to move forward, but the preclinical data will decide which one to select. And based on the different properties of these compounds, different indications can be selected to as potential to move forward into clinical trials. We also have the other series. This is different from the GT002, and this series have also very interesting properties that differ from the GT002 properties. And so we have a vast we have several compounds that we are actually characterizing currently.
And just to chip in there, I can just say that, as Stefan says, some of them are actually NAMs, so that means that they're negative allosteric modulators, and that means then that we even have potential with some of our compounds, very different indications such as stroke.
Exactly. And also epilepsy. So the refractory epileptic, this is also an indication that potentially could be explored with this type of compounds. So, I mean, we have huge possibilities with our pipeline compounds.
Yeah, exactly. And I since everybody wants to, I also want to chip in. And my chip in would be that remember that, you know, that the selection of GT002 as the horse we put all our money on is like 5-6 years ago. And in the preclinical work that we have done in our lab, it turns out that the other compounds, not only are they more interesting, they might actually be easier to work with too. But that was when the horses had left the starting point. And we had everything on GT002, but the potential in the pipeline compounds, and I really appreciate that question, is actually there, and it might be actually easier once we've broken the ice with GT002. Remember that the molecular structure is very similar between that pattern group and GT002. So there's definitely something there, and that is why we always mention our pipeline in all our presentation. But it's nice that somebody is actually interested in our follow-up little children.
Yeah, absolutely. And I think also, I mean, in a sense, what we have done, we have in a sense struggled a bit with the formulation, development of the formulation for GT002. But this formulation can potentially also be used for the follow-on compounds because the structures are similar and the properties are similar. So this is sort of an advantage that could speed up the path towards clinical trials.
Very good. Another question is, what makes you think that you can succeed in this very difficult area?
I think if I should start is that we have a very experienced team and we have a strong background in drug discovery development. We have been working more than 25 years in drug discovery and development in big pharma, in small biotech companies, in startups, and we know the opportunities. We see the opportunities when they come, and I think this is usually you don't give up. This is what you say, because big pharma, in a sense, they give up when they don't see the properties of the compound that they actually have developed, and then they go forward to another. But I think we are different because we see opportunities all the way, and we are stubborn and we are determined to take these compounds to the clinic and to the patients. I don't know if you want
questions.
I absolutely agree with Stefan, but just one thing I just maybe want to also, I mean, I agree 100%. You can make a lot with a good team, and it doesn't matter if you have a very good project and a bad team, you won't succeed. So that's one of the things they absolutely think. And of course, that remember that we control most of the drug development from the molecule to the clinic ourselves. So that is also an absolutely huge advantage. And if you look at the man hours that Christine, Stefan, and I have spent in drug discovery, you almost get to 100 man or woman years, which is actually quite a lot. So it's not the experience, and it is definitively, that is undoubtedly a very challenging project. And maybe perhaps to go back also to advance, all, as you can hear, all three of us have our background in the sciences, which is of course a very big advantage in the sense that we know what we're talking about, but it's of course perhaps a disadvantage because we're very conservative with what we want to actually go out and tell people before we are 150% sure that what you're saying holds and is actually true. And that comes with training in the sciences, I think.
Yes, and I'll just chip in here too, and I can certainly say that given that I worked at Astra and AstraZeneca, particularly within the CNS, and of course, big pharma such as AstraZeneca doesn't actually work within CNS anymore. So this whole area was dropped because in terms of, trying to get a blockbuster, then of course it worked much better for AstraZeneca to be within other therapeutic areas. And very definitely within CNS, I can definitely say that, well, we did have a drug for schizophrenia within CNS. It had to be withdrawn from the market because unfortunately it had very serious adverse effects and patients died. And that drug can still be prescribed, but only in Sweden and only for certain psychiatrists. So I have seen again and again and again compounds that have gone down at different phases. Some companies, some drugs have even been withdrawn from the market. So it's quite true that it's a very difficult field to be in, but at the same time, we have a potential drug that has a rather different mechanism of action, and we hope that it will succeed. But there's no guarantees, of course, absolutely.
Okay, we got one more question here in the meeting that just got in, saying there are other potential compounds coming up to provide pain relief and other benefits in the central nervous system with fewer side effects than benzodiazepines, i.e. Valium, which also hits the GABA receptors indiscriminately. Are there other competitors that you would say that you are comparing yourselves to?
Well, if I can start the chip-in session, I would say not really, because our compound, although it targets the GABA-A receptor, completely different molecular structure. It does not have the same non-selectivity as the benzodiazepines. It's actually a rather selective one. And we have not actually explored the entire area of pain, which is another indication which we have not put on our list, which is definitely something where there's a potential there. But in the priority, when we did the initial assessment of our drugs, we did not include pain models. They're not particularly nice to work with. And the other models seem to be much, much more interesting. And if you look at the modulation of cognition, I mean, there's a huge potential there. So we've very much focused on that. And for that, we don't really have any, there isn't really any compound we can directly compare ourselves. And also, you know, a lot of this focus within cognition has been the antibody field, and we are still in small molecular weight compounds, classical medicines.
Very good. We have one final question, so I'll give everyone some time also to write any questions that you still haven't asked in the Q&A section. But in the meantime, we'll just cover quickly the rights issue. There's a question here: why are you carrying out a rights issue? To start with.
Why we are?
Yes, just the reason for it.
We just to extend the runway for Gabather for continuing the activity, getting the agreement in place in order to finalize the clinical trial and start the clinical trial in patients too.
Very good. If there aren't any other questions, I can't see any others in the chat, and we haven't received any others beforehand. So thank you everyone for joining this session. This will be recorded, so you'll have a recorded version shortly as well. Are there any final words from you before we thank everyone?
Yeah, actually, I cannot stop myself from chipping in. I don't want to be the last one to be chipping in, but I just want to say that that the data that we've presented today, we're at the very beginning of actually going through this mountain of data that we have, and we definitely will have other presentations in the future where we condense this information and make it more of a tabulated data presentation than we have today. But the data that we had today, we also looked at it with the eyes of from an IP point of view, what we have looked at it yesterday, and also an idea in the point of view, and that goes back to big pharma. What the partners actually will want to see sooner or later, probably rather sooner than later, is a scientific presentation of that exactly data. So we have to be a little bit conservative with what we show, because the data has to be, of course, unknown if it gets your interest for IP or presentations.
Very good. With that, thank you everyone.
Thank you.
Thank you.
Thank you.